Our next presentation is Kriya Therapeutics, and I'd like to welcome Kurt Herberts, President and Chief Operations Officer, to the stage.
Hello, everyone. Kurt Herberts, President and COO, and happy to present on behalf of Kriya. And thank you to ARM and Meeting on the Mesa for having us. So, as many of you are aware, there's now 6 FDA-approved AAV gene therapy products. One of these is a subretinal delivery, Luxturna, and the other 5 are IV systemic infusions on the order of E13 VGs per kg to E14 VGs per kg, and all of them are for rare diseases with very high price points. So what Kriya is focused on is really leveraging AAV as a delivery modality, but for more common diseases, and we are not doing any systemic delivery. Everything is focally delivered into different tissues. And so this creates a new paradigm for gene therapy. Instead of focusing on tens of thousands of patients or 20, 30,000, it's really focused on hundreds of thousands to millions of patients with focal delivery, low dose, low cost of goods, and a much more reasonable price point. So here's the leadership team, a variety of pioneers in terms of operating discipline, clinical research, and complex biologics, as well as historical gene therapy expertise. The company was founded in 2019 by Shankar Ramaswamy, as well as Fraser Wright, our Chief Gene Therapy Officer. We've been highly successful at raising capital and committed partners to help us on this mission. Patient Square is the largest shareholder, and to date, we've raised over $600 million to focus on in-house programs that we can move into phase 1/2 clinical studies. There's 3 core principles that we have internally in terms of our product development. One is computational biology that helps us actually design and optimize the genes of interest. The second is translational research to move these programs from the non-clinical studies into the clinic. And the third is scalable manufacturing, both in terms of GMP manufacturing in-house, as well as an early-stage manufacturability assessment that helps us select the final development candidate that we make sure can be packaged, expressed, is functional, and can actually scale as we move towards the larger GMP activities. The majority of the company is in North Carolina. We have 2 main sites there. One is an R&D facility on the order of about 25,000 square feet with an embedded vector core that uses very similar upstream and downstream processes to what we're doing at the GMP scale. And then what you see here is a picture of our GMP site. It's about 51,000 square feet, 2 GMP production suites, and we can do GMP 50-liter, 500-liter, and 3,000-liter scale, which is essentially a commercial scale. Everything is upstream suspension 293 process and downstream column chromatography that is highly scalable across different serotypes and different transgenes of interest. Additionally, we have embedded in-house process development as well as deep analytical characterization for all of our programs, as well as QC, so we release the product internally. I should mention this facility is also QP compliant, and that allows us to go globally in terms of the phase 1/2 clinical studies, and the plan is to go globally for the pivotal studies as well. So when we think about product development, we really think about the protein. And then ultimately, we have to vectorize this from an AAV gene therapy perspective. But on the left, this just gives you a couple of examples of what's in our pipeline. Peptides, enzymes, monoclonal antibodies, fusion proteins, and engineered ion channels. And these have different utility, both in terms of the route of administration, as well as the particular indications that we're going after. And then there's a intelligent vector engineering step to think about how do we actually optimize that protein, encode that into the AAV, develop the optimized gene of interest, make sure that it's packaged, it's functional, can be expressed, and gets to the site of disease. So this slide I love because I start on the right-hand side, and we internally have integrated a lot of the cross-functional disciplines to focus on, first, a commercial product profile. And what this looks like is everything across the board in terms of the route of administration, the site of delivery, what type of physician or procedure is going to be used to actually deliver this to the patient, the primary endpoints, secondary endpoints, safety database, overall durability of a potential gene therapy product. And then we go back to the left and again focus on the protein, the vector genome design, the capsid selection, the promoter manufacturing evaluation, the route of administration, including plus or minus a device, as well as the in vivo testing. Once we finalize that development candidate, that then gets shipped out for the final GMP plasmid, and then we start all of the GMP manufacturing activities in addition to the IND-enabling studies with potentially definitive pharmacology and the toxicology studies. So our pipeline is unique compared to a number of other gene therapy companies, and there's really 4 key metrics that we look at. Number 1, as I mentioned, is direct-to-tissue delivery, and this is to get the AAV to the particular organs and cell types of interest and to minimize the dose. Number 2 is validated biology, so we're taking the risk on the gene therapy aspect as compared to a novel target. Number 3 is a de-risk development path, so we have a clear understanding of where we're headed based on the overall development path for other non-gene therapy products that have come before us in terms of primary, secondary endpoints, safety, durability, manufacturing, et cetera. And the 4th aspect is large commercial opportunities. And this is in terms of the prevalent and the incident patient population, but also the potential pricing that a gene therapy could command in this competitive field, agnostic to the therapeutic modality. And here's our pipeline. We have 3 main therapeutic areas of interest that we've decided to focus on. Number 1 is ophthalmology, number 2 is metabolic disease, and number 3 is neurology. For the purpose of this 15-minute presentation, I'll be going through 4 programs. All 4 of these programs will be going into the clinic over the next 16 months. So the first program here is in geographic atrophy. And just to tell a brief bit of the backstory, a couple of years ago, there were 0 approved drugs in late-stage dry AMD known as geographic atrophy. Now there's 2 approved products. One is Syfovre from Apellis. The other is Izervay from Iveric. Iveric has subsequently been acquired by Astellas. Both of these products are once a month or every other month intravitreal injection. And they have demonstrated the way to get 2 drugs approved in this indication, so patients now have an option. But we view them as kind of the first piece of the puzzle in terms of what this looks like for a therapy for these patients. So they have about a 20% reduction in lesion growth over time, and to date have not prospectively shown any type of visual acuity benefit for patients. And so this slide really articulates, on the left-hand side, our route of administration, and on the right-hand side, our mechanism. So the route that we're taking here is actually a suprachoroidal delivery, which is different from a lot of other gene therapies that have focused on subretinal or intravitreal administration. The suprachoroidal delivery we like because we believe we get better access to the choroid, the RPE layers, and the photoreceptor layers at the back of the eye. And we've completed a device collaboration with a group called Everads. And this is a novel device that has a microtissue separator that allows for better transduction and expression at the retina. We're using AAV2 here, and we did compete this against other novel engineered serotypes, all manufactured in our hands, and saw AAV2 as the best serotype to take forward for this clinical stage program. On the right-hand side, the payload, there's a number of other gene therapy companies that are going after more orthogonal targets within the complement cascade. Syfovre addresses C3, Izervay goes after C5. What we've developed is a CR2, CR1 fusion protein, where the CR1 component really inhibits the C3 and the C5 convertases, and so it's validated biology there. And the CR2 component holds that mechanism to the cell surface for those cells that are presenting complement deposits, so we want that therapy at the site of disease. Let me go back just a second. So this program is going to be filing a CTA this quarter and moving into dose escalation and dose expansion studies next year. Our second program is in thyroid eye disease, and the backstory here is Tepezza was Horizon's main drug. Horizon was acquired by Amgen a couple of years ago for $28 billion. Tepezza accounted for about 51% of the revenue at the time of Amgen's acquisition of Horizon. And one interesting story about Tepezza is it was originally developed as an oncology drug, so it's dosed as an oncology drug, sometimes is an IV infusion every 3 weeks for 8 infusions, in-office infusions. And the drug works pretty well. Physicians are pretty happy with the way that it works from an efficacy standpoint. But after that 6 months of administration, about half of the patients tend to have a proptosis rebound rate because they're no longer receiving treatment, so there's a loss of efficacy. And then the IGF-1R mechanism goes everywhere for Tepezza and causes on-target but off-tissue side effects in the form of hearing loss, muscle spasms, and hyperglycemia. And so that was essentially the problem that we were trying to solve with the gene therapy solution. So what we've developed is an AAV that encodes for an IGF-1R antibody that inhibits IGF-1R in the same way as Tepezza, but it's locally dosed. And so we inject this via a peribulbar injection, which is a common injection for oculoplastics, and basically transduces the muscle and the fat behind the eye and locally expresses an IGF-1R antibody to achieve the therapeutic efficacy over a multi-year durability, but to have very low systemic expression of IGF-1R, thereby hopefully minimizing the toxicities. That program, the thyroid eye disease program, will be filing a CTA or IND in the first half of 2025. The next program is in type 1 diabetes, and this is a very novel mechanism. The dosing is via intramuscular administration of an AAV1 vector. It's actually 2 separate genes of interest that are manufactured separately but pulled into the same syringe at a fixed ratio of insulin to glucokinase. The desire for insulin is to have both an autocrine and a paracrine activity and is dosed to be expressed at subbasal levels of insulin in the body. And the glucokinase is an intracellular protein that will exist inside the transduced muscle cells and help to metabolize the glucose. So the image here on the right-hand side shows the mechanism. It shows insulin being secreted from the muscle cells and then also coming back into the insulin receptor, which helps GLUT4 come to the cell surface. GLUT4 is a glucose transporter within the muscle, and muscle normally metabolizes about 80% of glucose in the body, and so we're basically helping the muscle metabolize the glucose even better with the glucokinase. In the muscle, normally, there's an enzyme called hexokinase that metabolizes glucose at very low glucose levels, and glucokinase can metabolize a lot of glucose at high glucose levels, and this creates the dynamic response, even though these programs are under the constitutive promoter within the AAV. So this program, we in-licensed from Fatima Bosch. She originally had done a lot of work and published this in canines, going out 8 years, where they became completely insulin independent. We then optimized those constructs for humans and took it back into mouse studies, and we're right in the middle of the non-human primate dose escalation studies, the toxicology studies, and the manufacturing for this will happen later this year. The last program that I'll speak about is also an intramuscular administration. This is a single vector, AAV1, dosed into large muscle groups expressing FGF21. So there's 2 companies out there that are doing modified versions of FGF21. Akero and 89bio have once-a-week sub-Q injectables for MASH. They've demonstrated and released data in F2 and some preliminary data in the F4 patient population. And this is a particularly interesting program because FGF21 has demonstrated strong anti-fibrotic activity within MASH. And we are basically matching a lot of the expression levels in clinical data for Akero and 89bio, but with our non-clinical data with an AAV vectorized FGF21. That program will also be going into the clinic in the second half of 25. So in summary, thank you again for having us. What Kriya is focused on is really bringing AAV gene therapy to more common diseases and making gene therapy a mainstay of traditional medicine. Number 2, the team is highly experienced across both protein biologics, gene therapy, and overall building out organizations. We've been successful at raising capital to date, over $620 million in. We started the year with 325, which brings cash runway into late 2026. We've developed this fully integrated engine across all of the in-house capabilities, the non-clinical studies, GMP manufacturing, and we are QP compliant and looking globally for our product development plans. And the pipeline has 4 programs going into the clinic over the next 16 months, and we're excited to execute against the dose escalation and the dose expansion within these common disease indications. Thank you.